Спасибо большое. Good afternoon, everyone. Start collecting. I see that some people are waiting for a lecture, so we have a bit of delay due to some technical problems. Now it is solved already, so start collecting, people. Good afternoon, guys. Good afternoon. Rukaya, good afternoon. You are the first one here. Good afternoon, Lavina, and all good students. So, good afternoon. So, today we are going to discuss HIV. And I would like to remind you that first, HIV as the causative agent of the disease had been identified in 1983. So, they found the disease first and then the causative agent of this disease. And HIV, I hope all know, stand for human immunodeficiency virus. So that's what we discuss today. And before we get acquainted with the coronavirus, HIV virus was the top virus. So, and one of the most important problems connected with the HIV virus is we are studying it very intensively, starting from 1984. So how many years? So, but then you cannot imagine how much money and efforts had been spent for it. So, but then until now, we have not so bad treatment, thank God. But until now, we don't have protection against HIV. We don't have any vaccines against HIV. And even with our very good drugs, until now, we cannot eliminate it from the body. Once it enters the body, we may suppress the growth of the virus, but we don't have any chance to eliminate it yet. Of course, there are some efforts. We've heard about some efforts that may somehow target the T cells, but the problem is that even resting T cells, even teenage cells can be infected with HIV. That is the problem. So that's why the whole pool of T cells in the body supposed to be infected and possibly to be infected. So the problem is how to eliminate it from the body. Okay, we start with global statistic. You all know the global statistic for corona. And of course, corona is not that little and that deadly and to compare with HIV. So take a look at this statistic, please. So since 1983, when it had been discovered, till 2019, 75.7 million people have become infected with HIV. Then 32.7 to 33 million people have died from AIDS since 1983 till 2019. So it was estimated in 2019 that at that moment 38 million people live with HIV and 1.9 million people become newly infected with HIV every year. So, for example, this is for 2009. But here you can see a bit of improvement. If you compare with 2015, so they were 2.1 million people newly infected with this year. So at least we have some decrease over the newly infected people within these 4 years. Then we also have decrease in people who died from AIDS. That is the terminal stage of HIV. So now it is estimated as 690,000 dead from AIDS. And if you compare the 2019 and 2016, in 2016, 1.1 million had passed away from HIV, from AIDS, right? So there's almost half decrease over the lethality. So then, among children, so 1.7, these are adults newly infected, so it is not written here. And among children, we have 150,000 in 2019. And that's also half from those that had been infected in 2010. And this is due to many programs that are undertaken or settled in the by different anti-AIDS and anti-HIV foundations. And that is the very good job for WHO organization. So now let's take a look at the who are at higher risk of being infected with HIV. So this is something about very young people. So the risk is 29 times higher among people who inject drugs. I hope there are no drug intravenous drug users here. Then 26 times higher among gay men and other men who have sex with men. And you as doctors need to understand why. And I hope those who know histology and the structure of the epithelium for example, female genital tract, and the structure of the epithelium in colon, they understand that epithelium in colon can be damaged much more easier, and so micro injuries that can be result from the anal sex, then they can give the very good pass to the virus through this injury. So that's why we estimate the risk 26 times higher among gay men. Then, of course, this risk is 30 times higher for sex workers. That's understandable because they are very often exposed to the virus. And 13 times higher for transgender people. Okay, we go ahead. So what is very important? What changes can we see, or what trends can we see in global situation? So if we take a look at number of people living with HIV and accessing treatment globally, then you can compare. So in 230, so around 30 million people had been living with HIV, but the only 770,000 people had been treated with the antiretroviral drugs. So then, and you can see, and there's the blue, these are the people receiving treatment. So you can see the gradual increase of those people for whom the therapy start to be more available. And what we've got in 2016 is 49% increase in people that get access to the treatment. So out of 36.7 million, so 19.5 million had been treated. And the new results, this is 2019. So in 2019, 25.4 million people get access to the antiretroviral therapy. And that time it was even more than 50%. So that looks really optimistic. And the increase of the number of the people living with HIV is not only because newly infected cases, but also due to the more effective treatment. And you know that we may have highly effective antiretroviral therapy. So, and then it allows people to live longer, and the treatment also improve the quality of the or living standards of the people infected with HIV. And that is important very much. So now from the epidemiology and statistic, we go for retroviruses, because this is the large family that is called Retroviridae. Retroviruses briefly are there. So what are the common characteristics for all retroviruses? Of course, they are unique. So they are enveloped, positive strand RNA viruses with a unique morphology and means of replication. And the unique replicative cycle depends on the very important enzyme, unique enzyme, RNA dependent. Oh, sorry, that's A here. RNA dependent DNA polymerase. And it had been called reverse transcriptase because it gives possibility to the virus to replicate through the DNA intermediate. So when the DNA copy of the viral genome is produced by reverse transcriptase, and this DNA copy is then integrate with the host chromosome or into the host chromosome. My dears, it was discovered by Baltimore and Temin. You might know these names from biochemistry in 1970, when the first so-called retrovirus had been discovered. But it was not HIV, it was animal retrovirus. So you see the classification of the family Retroviridae. And here we have many retroviruses that may infect, or here mainly, that may infect species other than humans. And it appears that those retroviruses may cause diseases in poultry and other birds, in a lot of mammals, and sea in koala, and a lot. Feline viruses also found. So we are not going to discuss them very today. So, but then we may have delta retroviruses, and there we have human lymphotropic viruses from 1 to 4, simian T lymphotropic viruses from 1 to 4, and bovine leukemia virus. So these are oncogenic retroviruses. HTLV human viruses are called briefly HTLV. So, and apart from these retroviruses, in this same family, orthoretrovirinae, we have the so-called lentiviruses, where the human immunodeficiency viruses, and also feline and some other mammals are there. So they are non-oncogenic, that what we think by now. And in separate subfamily Spuma retrovirinae. We may have so-called Spuma virus, and this simian formivirus and human formivirus. So, apart from those that we mentioned, uh, this is a three subfamily that may give rise to human infection. So we have Spumavirinae, Oncornavirinae, and Lentivirinae. So we may have the so-called endogenous virus or human placenta virus. So human HIV viruses belong to the subfamily Lentivirinae. So come back, that is lentivirus. So this is a lentivirus. So few words about the relatedness of the viruses. So you see uh, the gene nucleo as a result of nucleotide sequences of the genome that may show the relatedness of the viruses from this group. So you can see that you can see here both endogenous and exogenous retroviruses of humans, right? So when the exogenous is HIV and 1 and 2, uh, they stay quite far from the others, right? So that's why they are placed in a separate group and separate subfamily of lentiviruses. And here you can see the design, the structure, the design of the genome of the virus. So then we can see HTLV1 and 2 here from delta herpes viruses, but they have different configuration. So, but they all are flanked with long terminal repeats that we have here. LRT is a common, but then still the number of open reading claims and the order of the gene transcription and translation can be different. Then we have gamma retroviruses, and quite far from the others also Spumavirinae. These are exogenous that are zoonotic, but also endogenous. This is the only pole. So then, and beta herpes viruses also no exogenous, but we have endogenous viruses. So few words, just very few words about every subfamily. Spumavirinae. So we don't know whether they have some clinical disease or not by now. We know that they can produce very specific vacuolization of the cells or so-called foamy cytopathological effect. But this effect is not so strong, so this virus is non-cytolytic. We don't get the disease yet. So then we have a group of so-called endogenous viruses. What does it mean, endogenous virus? It means that the nucleotide sequences of the genome of the HIV or other retroviruses can be seen as a part of the human genome, and it is shown that endogenous viruses may make up to 1% of the human chromosome. And by now we think this endogenous virus is ultimate parasite. It means it occurs in the body in an only form of DNA, in a genetic form, as a part of the human host DNA. So these viruses will be transmitted vertically from mother to child during the meiosis, and that's it. So we never seen any productive infection caused by these viruses. We have not seen yet the science of this genome translation. So that's why we call them ultimate parasites. So then we come up to the family Oncornavirinae, oncornaviruses. Onco, these are oncogenic RNA viruses. So this is the cancer-causing retroviruses. From the human retroviruses, these are HTLV1-2 and HTLV5. We are not sure about third and fourth. So they are called RNA tumor viruses. So they immortalize or transform the target cells. So many often coronaviruses express the genes that act as analogs of cellular growth controlling genes. So that's why they cause oncogenesis by their own genes that may change the expression of some genes and cause the cells proliferation and immortalization. And you can see here that the types of the oncoronal viruses and all retroviruses can be classified. So this is for all retroviruses. They can be classified according to the symmetry and the structure of the capsid and the nucleic acid inside. So, and you can see here different A, B, C, D types of the capsids. So, and C type, this is absolutely large and icosahedral. This is C type, typical for HTLV from oncornaviruses. But D type is typical for another family, Lentivirinae. So, and you can see here this D type of truncated corn-shaped capsid. So, from now we will be talking about Lentivirinae only. So, and I would like to underline that these viruses, including HIV virus, they are non-oncogenic, cytocidal, so it means due to the replication inside the cell, as a result of it, it can cause, and it usually causes, cell death. So these viruses are called slow viruses because they multiply quite slow. And they also can be associated with neurologic, but also immunosuppressive diseases. So, and they are really famous as the cause of slowly progressive chronic disease that is called HIV infection that result, and the terminal stage of this HIV infection is AIDS. So, and this abbreviation was primarily used in 1980s, and it describes as a disease of homosexual men, Haitians, heroin addict, and hemophilic. So this was disease of so-called 4-H club. So, but the disease was found among those people at the very beginning of the 80s. But then it was found that young, healthy, normal people start dying of absolutely benign opportunistic infections. They start dying of really not so pathogenic fungi, for example, like aspergillus or penicillium that are always around us. And a lot of people everyone come in contact with this, for example, the saprophytic fungi. And nobody cares, but those people start dying out of this 4-H club. They start dying out of these diseases. So that was a problem. So Toxoplasma gondii, that is not really dangerous for humans. So that can be transmitted from the from cat or pet cat. So then they start dying of it. And that's why from the very beginning the virus was called HIV, human immunodeficiency virus, because it was shown that these people start losing immunity, start losing protection against the environmental microbes. And this discovery happened in two independent laboratories. On May 1983, by Montagnier in Paris, Institute of Louis Pasteur, and he published a description of a known virus, but he named it LAI. And in 1984, Gallois, National Institute of Health in the United States, reported the isolation of the human immunodeficiency virus from patients with lymphadenopathy and AIDS. So, and each group gave their viruses different names because they did know this is the same virus. But then it was established that the viruses are same. Ian Montagnier and Galore produce a common communique. They decided, or they share the priority of the discovery of the HIV. But then, of course, Montagnier was the first one. So now let's take a look at the structure and replicative structure of the virus, the genome and replicative cycle of this virus. So you can see they are spherical envelope. So they are middle-sized viruses, 80 to 120 nanometer in diameter. And they have, again, I would like to draw your attention at the very specific D type of the core. This is a cylindrical capsid surrounded by the envelope. So, and of course, there are some glycoproteins on the surface of the envelope. So we have basically two types. So, and these two types of glycoproteins form one single type over the spike, made up of two proteins. GP120 on top, so, and GP41, GP stands for glycoprotein, and 41 and 120 is the molecular weight of these proteins. So, and this is a stalk. So they call it lollipop structure. This is a candy and the stalk. You can see the classical appearance of the core in the scanning electron microphotograph. It resembles the truncated cone. So, genome of the retrovirus. You can see that we have two identical positive sense single-stranded RNAs. So each of 9 to 10 kilobases. So we consider this genome as diploid, two identical copies. So there in the core you can see these small proteins. These are the enzymes, this reverse transcriptase, and also integrase enzyme is shown there. So, and we have a copy of the reverse transcriptase and several copies of the integrase per virion. So, and you also can see that two cellular tRNAs are bound with the RNA, and they act as primers for the reverse transcriptase enzyme. So, and they are based on the each copy of the viral genome. So, and also virus encoded protease is the enzyme incorporated into the virion. So you can see a better picture of the genome, and would like you draw your attention at the repeats that are called long terminal repeats that flank the genome. So these are very specific sequences that contain very important sequences because they contain the very important fragments of the genome. So these are fragments that act as a promoter when it is switched on, enhances, and other gene sequences important for binding various cellular transcriptional factors that may up or down regulate the transcription and translation of the virus. So there we can see the genes for structural proteins and enzymes, and also genes for non-structural proteins. So the structural genes are GAG, POL, and ENV or envelope. GAG, this is the gene that is called group-specific antigens. So this gene gives rise to the internal proteins of the virus, you can see here. So initially, this gene will be transcribed as mRNA that will be translated on the ribosome into polyprotein, P55, right? Polyprotein. And then by virus encoded protease, this polyprotein should be cleaved into the 4 functional protein. So matrix protein, nucleocapsid protein, but then I would like to draw your attention to the capsid protein. So this is a P24, the major protein that make the core. So this is important protein because usually if you would like to, if you want to know is there a lot of viruses multiplying in the organism, producing in the organism, we can make the ELISA assay to find the number of P24 molecules or the concentration of this capsid protein. And the concentration correlates with the viral, so-called viral load, the amount of the virus in the blood, for example. So then you can see the POL gene, also structural gene, from the word polymerase. So this gene encodes the polyprotein that when it will be cleaved, it will be cleaved into several fragments, and these fragments are enzymes. Reverse transcriptase, protease P10, so integrase P32, and RNase P15 altogether. The 4 proteins can be generated out of one long polyprotein. So, and then we have a last one. This is the envelope gene that will be transcribed into mRNA and translated into the very long polyprotein that has 160 molecular weight, 160 daltons, kilodaltons. So, and then it will be also proteolytically cleaved into GP41 and GP120 to make the viral glycoprotein that combine together to form spikes. So that's what we discussed already. Then we have accessory genes. Please take a look at them here. So these are VIF, VPR, REV, TAT, NEF. These are the major, and VPU instead of VPX. So we take a look at them. So there's a several, around 6, sometimes we think 7. So genes with the corresponding products, gene products. So, and they have very important functions. So these functions are regulation of the viral genome replication and the proteins production, and it is important in regulation of the stages of infection of HIV. Regulation from the productive stage to latent stage and back to the productive stage. So the most important one is transactivator of transcription. So why it is called transactivator? We mentioned it before. Because it activates not the only cellular genes transcription, but also, or not only viral genes transcription, but it also activates the cellular genes transcription. So it makes T cells to multiply. So, and the more targets for HIV, the better. So then the RAV also is important with the molecular weight 19. So it is important in regulation of RNA splicing and promotion of export to the cytoplasm. So for translation, that is important. So NAF is a very interesting gene that had been used for a long while as a target for gene therapy. So because it may induce the chemokine production by macrophages that they can activate resting cells. So that will result in AIDS progression, because the more T cells, as we said already, the more targets for HIV, the higher productive and multiplication of the HIV. So, and of course, there are several other genes, regulatory genes, that promote the viral assembly. This is important in morphogenesis of the virus and facilitate the release of the virus from the host cell, and also transport of complementary DNA to the nucleus of the cell and shut down uh, the cell mRNA transcription and translation. So this arrests the cell growth. So these are very important genes. So then let's take a look at the replication of HIV. So we said that GP120, this is the glycoprotein that determines the tissue tropism, because it coincides completely with the CD4 molecule. So, and here you can see the process, how the uh, virus binds to the cellular CD4, you can see here, but binding with the CD4 is not enough. So here we have the very specific region in the glycoprotein, in the GP120, that mediate the attachment to the CD4. And the major cells that keep CD4 on the surface are CD4 cells, and sometimes, under some conditions, macrophages lineage. But they have very few, and T cells have a lot of CD4 molecules. But the core receptor is needed for fusion. So it binds through CD4 and transmembrane core receptors. Usually they are called G-protein coupled chemokine receptors, CCR5 on the macrophages and CXCR4 on the T cells. So, and these receptors promote the most strict binding over the CD4 and then changing of the configuration of the spike and the expression of these GP41 sequences or amino acid sequences, fusion sequences. So the exposure them to the cellular plasma membrane. So, and then at this moment we have the, at this moment we have the process of fusion in between viral core viral envelope membrane with a cell membrane. So, and it mediates the viral entry. The membrane fusion occurs and the viral entry. So, my dears, it has been found that those people who have some mutations in the genes that code for these CCR5 or CXCR4 receptors, uh, they have been exposed many times for the HIV, so but they never get infected. So we understand that these core receptors are of utmost importance. So now we take a look at the replication of the virus. You can see that as a result of binding and of the CD4 and the CCR5 or CXCR4 core, so-called core receptors, the viral capsid enters the cytoplasm of the host cell. And then the capsid will be partially uncoated, and then the virus will enter nuclear membrane with the complete uncoating of the nucleic acid. So the nucleic acid will be released with the help of VPR, so into the nucleus. So, and then the pre-integration complex is formed, and then the viral will be produced, the DNA will be produced, and then DNA out of the viral genome will be integrated with the host chromosome. It may stay there in the form of latency. Can you see here? So this is the one way of the virus survival in the human cells. So it may undergo the latent stage. So, and it usually happens when TAT, activator of transcription, is not switched on, is not active. So then another event that may occur right there. So this will be the production of mRNAs, and you can see here. So this is the transcription process and the production of RNAs, and then the process of the uh, replication and translation of these mRNAs into the long polyproteins. And those polyproteins supposed to be cleaved. You see GP160 protein that will be cleaved into GP120 and GP41. And then these proteins will be transported to the cell membrane and expressed on the surface of the cell membrane. So that's usually happens with glycoproteins in the viruses. And then apart from this, the genomic or pre-genomic RNA will be produced out of the genome. And then this genomic RNA will be placed into the newly synthesized capsid. So, but the capsid will not be fully produced yet. The problem is that the cleavage of the capsid will be delayed. And when the nucleic acid is placed into the core will not be completely done. You can see here the M protein that will stay there. Then the uh, cooperation of the core proteins there under the membrane protein. So, and then we have budding process, but then you can see that the virion is not mature yet. So you cannot see the very typical the D-type core of the virus. So the maturation occurs when the virus already released from the host cell. So, and after the maturation occurs, you can see that infectious virion will be produced. So, with a very typical truncated core of the capsid. Then this virus or this infectious particle may perform for the next round of multiplication. So here a few words about the translation. As we said, that the structural genes will be translated as polyproteins, all then they supposed to be cleaved. So the release is by budding, and even in the scanning microphotograph, you can see that after the release, the virion from the cell. So this is non-mature. Can you see they are released, but we don't see the typical structure of the core. So, but then with some time, the core will be formed. So we can see mature HIV virions. So, and this is the picture that shows what is going on during the maturation, right? So they have the cleavage of the polyproteins into several proteins, so they form the uh, very typical capsid, right? Matrix form the line from inside, and that's maybe the interaction between the lipid membrane or envelope and the core. So then we may see here the nucleocapsid proteins that bind tightly with the nucleic acid to identical RNA molecule, right? And you can see the so-called tRNA taken from the host cell. So, and then you have a large protein. This is the reverse transcriptase and some other enzymes. So, and you also may see that some host receptors, see here, that is the virion, but still some host receptors will be seen on the surface of this virion, infectious virus particle, because it is taken from the host membrane. So, and because the virus have the fusion peptide, so the HIV is known to spread directly from cell to cell. That's why the CPE that is caused by this virus is the multinucleated giant cells formation. We can see it here in the immunofluorescent microscope, or syncytia formation, where a lot of cells fuse together because the infected, HIV infected cell may fuse with neighboring non-infected cell. Of course, when those many T cells fuse together, can you imagine how many new offspring virions will be produced by this in situ? And of course, the fate of those cells is the death very um, quickly. For example, in situ can be formed within 3 days of the infected cells incubation, and then after these 3 days they start dying very quickly. So this will be cytolytic infection, and you can see here. So the release of the new virus particles out of the cell. So one of the serious problems that we have with HIV is genetic divergence. So the divergent population of viral genomes comprises a spectrum of non-identical bi-related HIV genomes. What does it mean in just words? So it means that as soon as HIV virus infects a particular individual, within a month in this individual, the pool of viruses of different but related types will be produced, and they will all have some differences in genomes. So, and usually around these viruses and their genomes, we may have one wild type. So there is a major type of the virus with the identical genome structure. And then we also have some minor types of the viruses with the differences in genome. So why it is so? This because, is it written here? Okay. This because the reverse transcriptase is prone to make mistakes when it performs transcription. So, and those mistakes will be not fixed. So, and a lot of viruses with the mutated genome, as a result, will survive in the body. But then why it is important? So it is also important, for example, when we start antiviral treatment and when we try to choose the drug that will be effective in this particular patient, we really develop or test the best drug that can affect the major virus in this pool with a particular type of genome. So, but then, if we adjust treatment very well, so if we stop replication of this particular wild type of the virus, that is a major type in the patient's body. But then some viruses that distinguishes a bit from that one, they will survive this treatment because the major treatment was not directed to the minor subtypes of the virus in one particular individual. So that's why we stop replication of the major type of the virus. So we are happy and the patient is happy. But within next 1-2 months, the new generation of mutated virus will rise and it will be one more different type now of the wild virus that will be a major type in the body. So this is very difficult to fight with. So that's why when we start treatment of the patient, we have to reconsider the treatment quite often. So every month we have to check whether the drug that we prescribe to this patient is still working or it is not working anymore. That's why usually when we perform treatment, we use more than one type of the drug. So to target as many specific enzymes, virus-specific enzymes as possible. So to down regulate or to inhibit completely the replication of the virus. Otherwise, the new minor types will arise and start multiplying. So, and the new wild type will be formed in the body. So, it will escape from the treatment. But by the same mechanism, so they escape from the antibodies because the usually the regions of greatest divergence are localized to the envelope gene that codes for surface glycoprotein GP120 and GP41. The divergence in the envelope of the HIV complicates the development of effective vaccine because as soon as we develop vaccine against and develop antibodies against particular type of the glycoprotein. But then as a result of the genetic variations, So the configuration and structure of the surface glycoproteins will be changed. And if they escape from antibody response, those viruses have priority to multiply in the body. And those that are neutralized by the antibodies, they will be eliminated. But then the new type will be formed and arise in the body from those who escape the neutralizing antibodies. So, and we know that GP120 has five variable regions, and these regions um, diverge among the HIV isolates. They are different, and usually the most important antigenic determinant are in the so-called V3 region of the GP120. So because there we have a site for neutralizing, so-called neutralizing antibodies. Okay, now let's take a look at the pathogenesis of the virus. So you might know already, of course, that this is lymphotropic virus. So it has very high tropism for T cells because they are the cells that keeps many CD4 molecules and they express the CD4 molecules. So, and usually we call them T tropic or T lymphotropic. But also we may have a variant that infects macrophages very often. So we call them M tropic. But then the result of the infection in T cells and macrophages are different. Usually in T cells, HIV causes productive infection, syncytial inflammation, and the death of the cells. But because the macrophages may express very few CD4 molecules, the infection occurs very slow, the multiplication occurs very slow in the macrophages. Usually CPE, like syncytial inflammation, is never seen in macrophages. So, but then the macrophages serve as a reservoir for HIV viruses. So instead of killing them, they keep them alive inside and hide them. So, and of course, because of the infection of the T cells and T cells depletion during the disease, so HIV induces very important immunosuppression. So, and when immunosuppression is formed, then really we comes to the AIDS, the terminal stage of HIV infection. So the reduction of the T cell numbers will be followed by decimatation of the T helper function of the immune response and also the development of T delay type hypersensitivity reaction. So, and here I would like to show you the importance of some innate immunity cells that participate in the HIV infection. And you see here that the uh, usually the virus quite often naturally pass through the epithelium and it will be anchored with the toll-like receptors, sort of this toll-like receptors. So by the dendritic cells that internalize the virus and migrate to the lymph nodes to transfer the virus to CD4 positive lymphocytes. In immature Langerhans cells express langerine, which promotes viral degradation in Briebig granules. And then we have, that is the dendritic cells, and then the virus is taken to the lymph nodes, and these are dendritic cells. So the macrophages also can bind it and internalize it and recognize through TLR receptors of macrophages will be activated also. But while the dendritic cells play as those that bring the virus to the lymph nodes and really infect the T cells uh, with the virus. Langerhans, macrophages usually more so as a reservoir for it. So they may take up HIV by mannose receptor. So, and they keep it as a reservoir, and by the way, it may travel through the body, and the macrophages may take the virus into any part of the body. Okay, pathogenesis. Initially, the virus is uptaken by macrophages in the body, and then macrophage lineage cells become persistently infection. So it means the virus multiply there at very low level, but then the viruses can be shed. So then circulating macrophages, microglial cells of the brains, and alveolar macrophages and dendritic cells are all those that may keep the virus and spread the virus, and mainly dendritic cells contribute to the infection of the T cells, and then so they all together may contribute to the clinical manifestation of the disease. So and then T cell tropic HIV strains will be developed in T cells, and they will be involved later on in the infection. And you can see here the T-tropic HIV strain recognize CD4 and fusion with CXCR4 chemokine receptors may result in the HIV, that not infected HIV infected T lymphocyte, and you can see here a lot of virion that start budding out of one single lymphocyte. So, it causes CP that kills the host T cells by several mechanisms. So first, if we come back to replicative cycle, there's much more DNA copies will be produced in the cell, and the only one copy usually integrate with human genome. So we can see here where we are. So we can see there the accumulation of the, they are not shown, but we have accumulation in the nucleus, the complementary DNA copies. Why complementary? Because they are complementary to the viral genome. First, single-stranded DNA copy will be formed based on the genomic RNA as a template, and then double-stranded copy. So that is called complementary DNA or cDNA. It means it's a full DNA copy of the viral genomic RNA. So and then accumulation of these copies, cDNA copies of the viral genome in the cell, interfere with the normal physiology of the cell, or they disturb the cytoskeleton of the cell. Of course, it also accounts for the T cell death. So also, can you imagine if we have bonding over the viruses from the T cell membrane? Of course, the membrane, due to a lot of envelopes are made for the viruses, it starts to be more permeable, and it also allow it to be damaged by the osmotic lysis. Then syncytial formation is highly cytocidal, and also the viral infection may induce apoptosis of the cells. So, the ability of HIV to kill target cells, in our case, these are mainly T cells, correlates with the amount of CD4 molecules and expressed by T lymphocyte. Here, my dear, I would like to stop to discuss some practical implementation of this thesis. So, my dears, so the major targets are T cells. So, the more T cells in the body, the more targets for the virus. So, the higher the production of the newly synthesized virions. But then I want to draw your attention that if you want to perform vaccination or immunization of the patient, when we, or any person, so when we inject the antigen into the body, we activate the chain where the macrophages will be activated, and the macrophages send the information to the T cells. So when T cells start producing more CD4 molecules on their surface, so when they are activated, but the more CD4 cells they express on their surface, the higher risk of getting infected with HIV for this particular cell. So that's why when a lot of people appeared with the immunodeficiency, HIV-mediated immunodeficiency, the question raised whether we can or we should vaccinate those people to give them protection against other human diseases or not. And until now, this is considered to be really very tricky and very dangerous to vaccinate the people HIV with HIV infection. Because if we decrease the number of the virus as soon as we inject a new antigen, we may activate the activation and proliferation of T cells. So then again the virus can start very fast multiplication in the T cells. So, and there's a few words about the immunopathology, and you can see here the binding or the entry of the virus through CCR5 receptor in the cell. And then this process may activate the expression of FAS and HIV-specific T cytotoxic lymphocytes will be directed to kill the infected cells. This is one of the also important mechanism over the T cell depletion, because a lot of T cytotoxic lymphocytes will be sensitized in the patient body against the viral glycoprotein that can be expressed on the surface. So then, or the FAS protein that will be expressed on the surface. So these will be the targets for HIV specific killer cells to destroy this. So, and by this route, the way of apoptosis will be activated, right? So we know that there's a procaspase 8 will be developed into caspase, and through cascade mechanism, caspase 3 will send the signal into the for the fragmentation of the DNA molecules inside. So then, some more activation pathways can be used. So, for example, the TCR of the lymphocytes also may recognize some proteins, especially the product of the NEF gene that is called NEF product will bind with the TCR, and it is quite immunogenic. And then the signaling mechanism will be activated and transduced into the cell to produce the signal for mitochondria-mediated apoptosis. So there are several ways how the activation of the T cell death can be performed. So then, how the virus usually behave itself and spread within the body? So the HIV infect macrophages and cause their persistent infection. And the persistent infection and the macrophages will migrate in the body, but then as a result of productive infection, it will shed newly synthesized viruses, and it also may take it to the brain because macrophages travel everywhere. So it is very common that it may cause neuropsychiatric manifestation of the disease. So this is the demonstration. This is a computer tomography of the brain in the patient with AIDS. This is a normal CT scan, but this is the scan from the same patient 4 months later when he presented with the mental slowing and confusions. And you can see the profound loss of the brain substance and enlargement of the fluid field ventricles, and that is the sign of the neurological disorder. So then the T lymphocytes will be infected. So first they cause persistent infection and then highly productive infection. As a result, we have immunosuppression. That is the problem. The virus kills the T cells, but virus doesn't kill the host. So, because the virus doesn't really target the very important organs, but then as a result of the immunosuppression, the disease will be developed. That is just opportunistic infections and neoplasia. So, neoplasms development. Why? Because uh, if we have the depletion of T cells and their function are T helper cells, I would like to remind you. So, but those T helper cells are important in both humoral and cell-mediated immune response development. And cell-mediated immune response and T cytotoxic lymphocytes activation is the major mechanism that protects us against tumor formation. So that's why the risk of development of neoplasms development is much higher in immunosuppressed HIV infected individuals than in normal people. So let's take a look at the cause of the disease. This is usually long-term chronic disease, and the cause is in parallel with the CD4 numbers. You can see them on graph. So the red one is number of CD4s in the blood. So, and if you take a look at this picture initially, so we have a rise in the virus in the blood. So it can be measured, as I said, by the amount of P24 core antigen in the blood. So if it is high, so it means the virus is multiplying in the blood, exists in the blood. So that we see very high, but then the virus will disappear from the blood during the second stage. First stage is acute infection. Second stage is the stage of chronic lymphoadenopathy. The first acute infection usually uh, uh, lasts for several months up to a half of the year. So then it starts to be the chronic lymphoadenopathy stage, when the virus usually is hidden in the lymph nodes, in the T cells. Of course, it multiplied there, but because the antibodies are raised, the titers are raised in the body already, of course, because a lot of viral antigens cause the stimulation of the immune system and the development of the antibodies. So, the rise in antibodies usually coincide with the elimination of the virus from the blood. So that's why, as far as antibodies are high, so the virus is not seen in the blood. So usually it is located and it is hidden in the lymph nodes, and from there it will be produced. So then the next stage that follows the chronic lymphadenopathy, this is subclinical immune dysfunction. So sometimes this is the time course of the disease is divided into 3 stages or 4 stages. It doesn't matter. It's important to understand what is going on within those stages. So this is the subclinical immune dysfunction. And then it progressed to skin and mucous membranes immune defects that may result in opportunistic infection, start of the opportunistic infection. So, but then when the CD4 cells drop dramatically, then we have the progression into the systemic immune deficiency that is known as AIDS. So, and the way of the CD4 cell counts, we may have some milestones. So, when it drops down to 400 to 450 cells per ml, T cells per ml. So we are talking about the start of opportunistic infections. And when it drops down to 200, so we are talking about the development of AIDS already. So you can see these three important markers: the virus in the blood, the number of CD4 T cells, and the level of the titer of the antibodies. So usually describe and may be used to see in what stage of the time course of the disease we have the patient. Okay. Так. Transmission. HIV is present in the blood, semen, and vaginal secretion of the infected individuals. I hope you know this. And it may be spread through sexual contact and exposure to contaminated blood products or blood, and can be transmitted from mother to child. So we have discussed who are at risk right now already. And the first stages that we mentioned already, from up until the dramatic drop of the CD4 counts below 200. So all of those stages are called HIV infection. And the last stage is called AIDS. So what kind of symptoms do we see around the can be considered as clinical symptoms? Okay. Initially, so we just studied herpes viruses. Initially, it is mononucleosis-like symptoms. So, and or we also may call flu-like symptoms that subsides usually within 2-3 weeks. So they may be accompanied with a rash, meningitis. They also may appear or disappear. So the symptoms stem from immune responses, widespread infection of lymphoid cells. So then the asymptomatic period, when we have this period that is called the period of generalized lymphadenopathy. So then the stage of opportunistic infection starts when the opportunistic infection is really the result of the deterioration of the immune response. So usually the appearance of the symptoms correlates with CD4 counts. And at the end we have the full-blown AIDS, when again CD4 drops below 200 per ml, microliter, sorry. and more significant diseases appear there. So we take a look at them. What is very typical? So-called HIV or AIDS wasting syndrome with the lipidodystrophy in AIDS. You can see it, this lipidodystrophy here and there. Then was so we're talking about aspergillosis today. This is the X-ray examination with the developed, and what we see is aspergilloma in the chest. So this is a highly lethal disease for immunocompromised patients caused by this very uh, benign fungus, Aspergillus flavus or whatever other Aspergillus. So then the regular candida that are always with us because they are members of normal flora of the body. They may cause local but also very severe systemic infections. In immunocompromised HIV patients, it can cause sepsis, very severe oral candidiasis, and candidiasis of esophagus, trachea, bronchi, and lungs. So finally, it caused the death. Very severe fungal infections like sporotrichosis may occur in the patients with AIDS. That you see the result of it. Then, do you remember Kaposi sarcoma associated virus? So the Kaposi sarcoma is formed, so lesions are usually located on the gums or hard palate and can appear macular, nodularized, or ulceration with color range from red to purple. And that's because uh, not the HIV itself causes it, but the immunodepression make patients unable to find with the gamma herpes viruses. For example, EBV virus or Kaposi sarcoma associated virus. Then oral hairy leukoplakia of the tongue, Kaposi sarcoma once again. So if you take a look at this is a diagnostic testing algorithm for primary immunodeficiency disease. You can see how many different types of the signs should be considered uh, to develop the diagnosis according to the laboratory indexes. So what do we usually use in the laboratory diagnosis? So the estimation of viral endogenous blood is not in use anymore very often because it is considered not stable. So we can also perform virus culture, but it is of utmost danger. The very specific labs with a very high degree of the isolation from the environment according to the biosafety levels, 1 to 2 should be used. Then it is not really used in the clinical routine diagnosis of HIV infection. Then the reverse transcriptase can be identified by ELISA tests. So viral RNA can be found through the PCR test, and also serological tests are used. Then, if you first time come to the diagnostic lab to be sure whether you're infected or not, most probably the serological tests will be used. So ELISA, indirect ELISA to find antibodies, and this is used to document the HIV infection. So by one ELISA is not enough because it is not that specific enough. So it is used for so-called routine screening, but then because it may give false positive results. So if it is positive, then the document HIV infection should be confirmed by Western blot analysis because this is considered to be more specific procedure. Why? Because as a result of Western blot, you may demonstrate against which particular proteins of the virus the antibodies are produced. It is still test for antibodies to confirm the presence of anti-HIV antibodies in the blood. So there is a confirmation test. So, and as a result of this, the patient can be considered seropositive. What does it mean? So, when we took a look at the time course of infection, please take a look. So, this is the so-called serological window. So, why? Because the patient doesn't have antibody yet, but virus is already excreted. So it means the patient is infected, by antibodies are not produced yet. We may have this window period, so-called window period, without antibodies for up to half a year. So but then antibodies will appear in the blood, so it is a sign of conversion of the patient from being seronegative to seropositive when antibodies can be found in the body. So then we are not discussing culture because this is usually performed in very special reference labs. So, and then one more very good, by now maybe the best one, is the identification of the number RNA copies in the blood. So, and as a target sequence, we use target sequence for reverse transcriptase. So, so-called RT, reverse transcriptase PCR, is performed. Few words about treatment. So, my dears, so there's so many now targets for treatment. So when you can see here the picture where we can see the targets for treatment. Of course, the best one is reverse transcriptase inhibitors. So then, if we are talking about the early stages, the so-called binding and fusion inhibitors are already used. So you can see here the same picture. And so this for binding, we may use this monoclonal antibodies, MAB. Do you remember monoclonal antibodies to block the CD4? And we may use also the co-receptor binding inhibitors. So, that is the inhibition of the fusion. And this is Maraviroc and Vicliviroc, two preparations that are used. And the really fusion inhibitors, there's enfuvirtide. So these are the drugs that are used already. That's their description, a bit more for the description. So then, but of course, the major group of drugs are RT or reverse transcriptase inhibitors. So we may have nucleoside analogs and non-nucleoside analogs for this particular enzyme. Then also the protease inhibitors are developed. Do you remember the protease is responsible for proteolytic cleavage of polyproteins, interfunctional proteins. So core proteins, that is GAG, proteins that are enzymes, and ENV protein that are GP120 and GP41. So, and then the last one is integrase inhibitors that inhibit the integration of viral C complementary DNA into the human genome. So these are a lot of names of these, but then you may read them in the book. So protease, and these are the last group of drugs, this integrase inhibitor. So they are used in cocktails. So because this therapy should be very well adjusted to every single individual. So due to the problems of side effects that can be used, and also it is very important that these all drugs may be combined in one single pill. So this make the taken pills not so often, and that's also improve the quality of the patient life when not so many times you should take it a day. Okay, so we almost finished. So with this we have to finish, and because I have a group right within 10 minutes. So my dears, very good that you come, and please, we finish.